A viewer has asked about the television ads that are showing in the United States that are pushing the idea of 55 milligrams per deciliter, which is about 1.4 millimoles per liter, as the target for LDL for patients on secondary prevention. I decided to enlist the help of ChatGPT for this one. Stay tuned. I generally don't have a well-developed position when it comes to secondary prevention because the statistics and the circumstances are totally different than those for primary prevention. And that's why I decided to use ChatGPT for this one. It's an experiment and to see what I could learn. I have to admit, the experience sent me down a whole bunch of different directions and I'm only able to cover some of them. So let's look at the question. Pretty straightforward. What do you think about the latest TV commercial saying that we patients in secondary prevention need to get our LDL-C down below 55 milligrams per deciliter? I cannot find any rock-solid science that supports such a recommendation. So a few notes here. This viewer is in secondary prevention, meaning at the very least they have a known cardiovascular disease and possibly an actual event, a heart attack or a stroke. They're asking a very important question. What is the justification for this particular number, 55 milligrams per deciliter or 1.4 millimoles per liter? Why is that target being pushed? Why that and not some other number? So that's what I'm going to look at. I went to ChatGPT and I typed in this exactly except for the part with the 1.4 millimoles per liter. I just said, why is 55 milligrams per deciliter an LDL target? Now each time I typed that in, because I tried it a few times with the exact same words, I actually did get slightly different answers. So I'm just going to go with the first one that came up. And it gave me this answer. The target of 55 milligrams per deciliter for low-density lipoprotein cholesterol is recommended for high-risk individuals due to several reasons grounded in clinical research and guidelines. And then it broke up the discussion into these areas: association with cardiovascular risk, evidence from clinical trials, etc. And it went on also to have a section on practical implications and then a conclusion. So we're going to look into each one of these areas. Under association with cardiovascular risk, they had this statement: elevated LDL levels are strongly associated with an increased risk of atherosclerotic cardiovascular disease, including heart attacks and strokes. Lowering LDL reduces the risk of these events. So I want to point out something here. First, they say it's strongly associated. That means it's not necessarily causal. It's just there is an association. The LDL levels could be caused by the same thing that's causing the heart attacks and be a reflection of it, but not actually the cause. But then later they say lowering LDL reduces the risk of these events. Well, presumably they're saying lowering LDL with medications. And that adds a little bit more to this association word. It says that it is causal. I mean, I believe that statement implies that it's causal. And finally, this was the extent of the statement. No evidence is actually given here, but maybe we'll see it in follow-up sections. So under evidence from clinical trials, and this should be good because this is where we're going to see the evidence. They had this paragraph. Numerous clinical trials have shown that intensive lowering LDL-C leads to significant reductions in cardiovascular events. Studies like the Improve It, Fourier, and Odyssey outcome trials demonstrated that achieving very low LDL levels, even below 55 milligrams per deciliter, results in additional cardiovascular benefits without significant safety concerns. So let's dig into this a bit. First point I want to make is that all three of these studies mentioned were funded by pharmaceutical companies who stood to benefit from the success of the trials. So here's a pro tip. If you ever do get involved with a randomized control trial, try getting into one that is funded by the pharmaceutical companies because they always seem to do better for some reason. And of course you want the benefit from being involved in this trial. They're basically just that much more successful than independent trials. Now I'm not going to go into all the trials here. We will do a deep dive into the Improve It, and we'll look at some of the literature that I found. Briefly, the Fourier and the Odyssey outcome trials were for PCSK9 inhibitors, not statins, though that doesn't mean they're not pertinent to answer this question because the question really didn't say by statins. It's just asking us about the 55 milligrams per deciliter number. It's notable, though, that the Fourier trial, all-cause mortality was actually slightly higher in the treated group than in the control group. Not enough to be statistically significant. It could be just random chance. but there was no benefit that they could find, at least in all-cause mortality, and really that's what counts to us. I do want to point out this phrase, however, without significant safety concerns. These studies, of course, had a run-in period like all other studies, and among people who didn't have adverse effects, they managed to achieve low LDL levels, but that says nothing about people who were suffering from adverse effects because they typically dropped out of the trial. So this statement about safety lacks any meaningful context. So let's look at some of the papers that I found. This one is called Improve It. How much really is the improvement? It was in the American College of Cardiology journal. And they point out while the relative risk reduction of 6%, which is a hazard ratio of 0.94, would not generally be thought of as very impressive, Improve It did note a reasonable number needed to treat of 50 in a high-risk population to prevent cardiovascular disease event. Thus, such added therapy would seem to be a reasonable option for high-risk subjects. I have to kind of take exception to the word reasonable attached to number needed to treat of 50. Full disclosure, a couple things to note. This trial that they're referring to was comparing Simvastatin Ezetimibe combination treatment versus just Simvastatin alone. This is just looking for the incremental benefit that they might see by adding Zetia to the Simvastatin. That the number needed to treat was 50. NNTs are very tricky. We have to get the context of them. Without context, they don't mean anything. For example, this doesn't say how long the treatment would go on. A number needed to treat of 50 for a period of 1 month would actually be kind of good because there's not going to be a lot of events and it's saying it's preventing them. A number needed to treat of 50 over 10 years, to me, it's just not very impressive. Another paper for Vytorin, that is the product that was being tested in this trial, in Zetia, the benefits were too small for a label change. What had happened was Merck was seeking a label change that would push the cardiovascular benefits of their product and was trying to get the FDA to approve that label change. So one of the panelists, the Dr. Milton Packer at Baylor University Medical Center, said, "It is not robust. You blink and you miss it, and you wonder if you care or you don't care about this minuscule benefit." Though some of the panel members did disagree, like it says here, at least one said that the FDA's role is to determine whether their trial delivered statistically significant results. Whether it's clinically meaningful is a question for others, including the people paying for it. I have a little bit trouble with that sentiment, though I can see where they're coming from. Should the FDA be saying, no, that benefit isn't good enough, you're not allowed to have it? Maybe, maybe not. The idea that's coming forth here is that statistical significance and clinical significance are very different. Something can be statistically significant, but not really clinically significant at all. From the Mayo Clinic website, just discussing cholesterol levels in general, can your total cholesterol be too low? Doctors are still trying to find out about the connection between low cholesterol and health risks. There's no consensus, but LDL would be considered very low if it is less than 40 milligrams per deciliter, which is about 1 millimole per liter, a little bit more. So think about that. They're pushing towards 55 milligrams per deciliter while saying 40, which is not really all that much lower, would definitely be considered too low. So maybe there could be arguments that 50 is too low. There could be arguments that 55 is too low. And then I just wanted to point this out. For example, people with depression may have low cholesterol levels, and it's not certain whether that's causal or associative. But it's not been proved that lowering cholesterol with statin therapy causes depression. Well, it's not in the interest of any pharmaceutical company to fund any studies that are going to look for that. So I don't know how much studies have been done there. I should probably look around for them. The next section was guidelines from medical organizations. And this will be quick because we know that guidelines from medical organizations are nothing more than opinion. They may be opinion based on some data, but they really are opinion and value judgments. The European Society of Cardiology and the European Atherosclerosis Society, they recommend this 55 milligrams per deciliter. That's not evidence, it's just a recommendation. And the American College of Cardiology doesn't have a precise number, but they support intensive lowering of LDL in high-risk individuals. That's interesting because it's in high-risk individuals, meaning secondary prevention generally, not necessarily in primary prevention. Another topic that is covered is plaque stabilization and regression, and they have this to say, they being ChatGPT, achieving lower LDL levels is associated with stabilization and even regression of atherosclerotic plaques. This contributes to a lower risk of plaque rupture, which can lead to heart attacks and strokes. Well, this is a whole separate topic. Some doctors who don't prescribe statins for cholesterol lowering do believe that low doses of a statin, generally Crestor, may stabilize soft plaque, and that is a good thing. But if that's the case, it's a pleiotropic effect, an effect that is not what was intended by the drug. The drug is intended, when we're talking about statins or even PCSK9 inhibitors or Ezetimibe or any of the others, their intent is to lower cholesterol. If what they're really doing is stabilizing plaque, and that wasn't their intent, maybe lowering cholesterol should be considered the side effect. I'm not going to go into that anymore here because it's a whole separate discussion unto itself. Safety and tolerability. Well, this is what they had to say there. Modern lipid-lowering therapies, including high-intensity statins, Ezetimibe, and PCSK9 inhibitors, which are the three we've been talking about, have been shown to safely achieve and maintain LDL levels as low as 55 milligrams per deciliter or lower in many patients. I take exception to the idea that statins at least do this safely. Whether they do or not, this is not evidence that 55 milligrams per deciliter is correct. And I also want to point out that ChatGPT in its answers hasn't made any distinction between the different qualities of LDL. For example, it's not making a distinction between oxidized LDL, perhaps pattern B, or the LDL profile, which would be pattern A plus other characteristics that, for example, a lean mass hyperresponder might have. It just doesn't have that in here. ChatGPT went on to tell us about practical implications. For high-risk patients, for patients with a history of the disease, familial hypercholesterolemia, or multiple risk factors, achieving an LDL-C target of 55 milligrams per deciliter is particularly beneficial. I have to wonder on this one, why is 55 milligrams per deciliter particularly beneficial for somebody with familial hypercholesterolemia? I mean, shouldn't just getting a person with FH down to normal, why wouldn't that be the goal? So why is familial hypercholesterolemia being mentioned here in the context of the 55 milligrams per deciliter? It just seems to be a non sequitur. Then, under treatment strategies, to reach this target, high-intensity statin therapy is often recommended, potentially combined with other lipid-lowering agents. Well, yeah, that's a true statement. To reach the target, this is what is often recommended. This doesn't add anything as far as the evidence for the answer or for the 55 being the right number, but it's a true statement as far as it goes. So, in conclusion, this is what they had to say. The LDL target of 55 milligrams per deciliter, again 1.4 millimoles per liter, is based on a robust body of evidence indicating that lower LDL levels provide greater protection. I have to take exception to the word robust there. From consulting only what ChatGPT gave me, there wasn't any particular justification for the 55 milligrams per deciliter number as opposed to any other number, higher or lower, being the correct one, other than the idea of lower is better, which permeates the conventional wisdom, if we can use the word wisdom in that case. So I don't see anything specific here in what ChatGPT has told me that makes 55 the right answer. My closing thoughts on this: is there scientific evidence that 55 milligrams per deciliter is a legitimate target? Well, I didn't see it. The so-called evidence was actually very weak. The Improve It trial certainly didn't do anything to justify this particular number other than, in general, lower is better, which was a statement in it. And if they had said, well, this level was chosen because it gives a relative risk reduction of this or something like that, at least we'd have something to compare it to, be based on something, and we could argue it. But really, this 55 milligrams per deciliter number, this is a lot like the 7.5% 10-year risk that we get out of the guidelines that make us eligible for statins. It's a value judgment. Science can't tell you what is the right number in this case. How much risk is too much to bear? That's an individual decision. And the same thing here with this 55 milligrams per deciliter, we would have to look at the the evidence, such as it is, about how much lowering we get and what is the marginal benefit of 55 versus 60 versus 70 versus 100. These are the things that a patient needs to be told in order for them to make this value judgment. So the bottom line is there really isn't scientific justification for this number, 55 milligrams per deciliter. In particular, when it comes down to it, in the end, it's a value judgment. Now, a secondary thing that I get out of this is, how did ChatGPT do? Assuming the algorithm did its thing without human intervention other than the original programming, I think it did a pretty good job at parroting the pharmaceutical company conventional wisdom and gave me a lot of things to chase down. But the key here is I did have to chase them down to find out what they really said, because if I just took ChatGPT at its word, I'd say, yeah, there's your justification. But once I looked into the studies, well, it really wasn't a pure justification for that particular number. ChatGPT, it's not advanced enough to discover anything independent or to add new information to the discussion or to make nuanced distinctions like between an association and causality. It just didn't do that. It'll probably be able to someday, but it just didn't do it in this particular case. But you can be sure that if AI did start to point out all the issues with statins, then the mainstream medical profession would start throwing bombs at it and say, this is no good, this is nonsense, trust us, take your statin. So that's all I've got on this topic. If you appreciate this content, please like, share, subscribe, and comment on this topic or others you'd like me to cover. And if you haven't seen this video, I recommend you take a look at it now. Thanks for listening.